All right, so we were in the middle of talking about translation and taking this messenger RNA in blue here and converting it into a protein, which I've depicted here in orange. And it's basically three processes. The processes are initiation, elongation, and termination. Initiation is when we start the translation. So basically that's when the small ribosome binds to the messenger RNA, and that happens at that 5' cap. Elongation is when the ribosome works like it walks along this messenger RNA and it takes steps that are 3 base pairs long each time. So it's going to move one codon at a time across the messenger RNA, and each time it's going to move a codon, a new amino acid is going to come in to match that codon, and then once it's done and you've added that amino acid onto the peptide chain, then what it's going to do is it's going to continue to walk through. And once it gets to the end, it reaches a stop codon, and then the protein is released and the ribosome dissociates, and it gets regenerated and recycled so that it can start the next cycle of protein synthesis. So translation initiation is the first step that we need to talk about, and this is really what we regulate doing resistance exercise. So what happens here is translation initiation takes advantage of the fact that most eukaryotic or most mammalian messenger RNAs have a 5' cap that protects the 5' end, and it has that the poly-A tail that we talked about before, and here the poly-A binding protein can bind to that poly-A tail, and that's going to help it be translated, because if the poly-A binding protein can't bind, that tells us that the message isn't intact, and so you don't want to translate messages that are being degraded. So the way that initiation works is we're going to use this 5' cap as the landing spot for a protein called the cap binding protein. Not surprising, because it binds the cap. We also call this protein the eukaryotic initiation factor 4E. So 4E, or the cap binding protein, binds to the cap, and that's the first step of translation initiation, and that's really not overly regulated. What is regulated is how it associates with the rest of the complex. And it's regulated by this little protein that I've depicted in yellow here as the protein called the 4E binding protein. So this is a protein that binds to 4E, and it binds in a very specific sequence of this amino acids of this protein. And it's an important area because it's binding and it's preventing the association of another protein called EIF4G. And when we get an anabolic signal, like we eat leucine-rich protein or we do resistance exercise, what happens is that this 4E binding protein gets modified. You add a bunch of little things to it, and what it does is it changes shape. And when it changes shape, it can no longer bind to the cap binding protein. And so it moves away. And when it moves away, this protein EIF4G can come in and bind to that exact same docking site. And that's the first step to really starting translation, because you can see that 4G has all of these different little dimples in it, and one dimple fits the 4E binding protein, and that's the way that we know that this message is intact and we should translate it. And the other little dimple fits this protein called EIF3. And EIF3 is really important because what it does is it is the chaperone. So it's the chaperone protein for the small ribosomal subunit, or the 40S ribosome. And so what it does is when 3 binds to 4G, when it's doing that, it's bringing the 40S ribosome into place where it can start translating this messenger RNA. If you think back to those transcription factors, the cofactors, and that enzyme, the polymerase 2, in transcription, this structure looks very similar. So basically all we're doing is we're taking the trampoline, we're loading it up with this big, huge protein, 4G, and the likelihood that you're going to get the ribosome rolling into the middle of the trampoline where you've put all of this weight goes way up. And that means that this message is now going to be translated, or that it's going to start translating. And so when it starts translating, for the 40S binds, then the next step is the 60S to bind. And once that does, it's going to move along the message until it finds the transcription start site, or the translation start site, which is it's an ATG. So it's an AUG is the sequence, and that's a methionine, and that's where we're going to start translation. Once you start going and you start building this peptide chain that we see here that's going to become the protein, this orange chain here, what happens is each codon, the ribosome, has little pockets in it. And each codon is going to have a space within those pockets. The ribosome moves down, it binds to this area, and so we're just about to add this one CAG, which is glutamine, and that glutamine is now in the acceptor site, and then what's going to happen is this enzyme here, the elongation factor 2, is going to facilitate the movement of the ribosome, and when the ribosome moves, the change in shape of the ribosome causes the glutamate to be added to this growing peptide chain, and then the whole thing moves over. So you can see that here is that this green tRNA is now in the exit site. This pinkish one, the glycine, is now in the acceptor site, and the glutamine is in the middle site. So what's going to happen is this is just going to keep walking down this messenger RNA, adding one amino acid at a time, and it's going to keep going along that messenger RNA for as long as it takes to translate the whole message. When it then needs to come to a stop, you have a stop codon. Stop codon in here is UGA, UAA, or UAG. And what is interesting about the stop codon is all of the other codons have a tRNA that gets charged with the amino acid that corresponds to that codon. The UGA, UAA, and UAG, they don't have a tRNA that binds to that codon. So instead you get a release factor comes in, the ribosome moves, and it passes it to no other amino acid. And so because there's no amino acid here to pass this peptide chain to, the peptide chain is released, and that causes dissociation of the ribosome. And that's when we've terminated translation. And now what we have is we have this protein that's been fully synthesized, but it's kind of not in the shape that it's going to be. So what we have to do is we have to fold it up. And the folding is going to be facilitated, or it's going to be made possible by chaperone proteins again. And what happens is the chaperone proteins come in and they help it fold based on a few rules. And those rules are that you're going to try and minimize hydrophobic interaction. What that means is hydrophobic, afraid of water. So the amino acids that are hydrophobic are going to go into the middle of the protein, and the hydrophilic ones, the ones that like water, they're going to go onto the outside. So the hydrophobic regions come into the core of the protein. And then you're going to form intermolecular hydrogen bonds where a sulfur group on one amino acid is going to form a bond with a sulfur group on another. Or that's a sulfur bond. Hydrogen bond is when the hydrogens interact with negative ions, so a hydrogen is a positive charge, and it's going to interact with a negative charge. And that's really what we call van der Waals forces, where basically a positive charged amino acid and a negative charged amino acid are going to come close together. A negative and a negative are going to be forced apart, and a positive are going to be forced apart. So you're going to take into account the hydrophobicity, the fear of water. Those are going to go into the inside. And then the charge is really going to be important for determining the secondary structure. All right? So that's how we get from a message to a folded protein.